Hello, everybody. My name is Housam Eidi. I'm a postdoc fellow from Professor Christopher Shaw's lab at the University of British Columbia, Vancouver, in Canada. I will talk today about our preliminary data of our current study on the impact of the neonatal vaccination of neurodevelopment and immune inflammatory responses in mice. So the current assumption by or of the American FDA on the safety of vaccines is still questionable. Because first of all, the FDA considers the vaccines as biologics, but not as drugs, which enables them to faster track the vaccine for their adverse event within few days versus several years in the case of drugs. And we have as well the the issue of the scarcity of the vaccinated versus non-vaccinated experiments, and the problem of the placebo as well, which is in the majority of cases is another vaccine, a group of vaccine, or even adjuvant. And in the literature, we can, several studies mention that, repeated immunization will lead to autoimmunity even in genetically non-susceptible animals, and the administration of immune adjuvant can overcome the genetic resistance to autoimmunity. And they highlighted that tight connection between the development of the immune system and that of the central nervous system. So the early life immune challenges will lead to long-lasting abnormal cognitive and behavioral responses. And last year we have two epidemiological studies that highlighted the strong link between the vaccination and some neurodevelopmental disorders, such as the learning disability and autism. And we don't have to forget that the majority of vaccine formulations have a certain number of neurotoxicants, especially the aluminum, as my colleagues mentioned earlier, especially the aluminum. That is a huge problem in kids, and with the problem of the overdose, according to the guidelines of the FDA, not according to my guidelines. And we have to mention as well the non-declared contamination with micro and nanoparticles in these vaccine formulations. Yet, the current US vaccination schedule, which is considered one of the heaviest one in the world, is still assumed to be 100% safe. So, this current assumption on vaccine safety should be replaced by an adequate scientific evidence. So, our study aimed to investigate the impact of the combined US pediatric vaccination schedule on the developing central nervous system and the immune system in mice to prove our hypothesis that the pediatric vaccines can interfere with the normal brain development and trigger immune pro-inflammatory abnormalities affecting the central nervous system. So to this end, we use two cohorts of black cyst mice, and we have 4 experimental groups. The first one was injected with weight equivalent of the current US pediatric vaccination schedule for 0 to 18-month-old infant. The second one was injected with the equivalent as well of triple of this dose. We tried to do a positive control for this study, and the third one was injected as well with the equivalent of aluminum in the amount that we found in the current US pediatric vaccination in the same window of age. And the fourth one was the saline or control group. So after the animal breeding, we have two cohorts of pups. They were vaccinated according to the CDC schedule, which between 7 to 22 postnatal days, which corresponds to 0 to 18 months in kids. And then this first cohort has to be sacrificed by the 22 days. And before that, we have to do a pre-weaning behavior test for the both of these cohorts. And then we continued with the second cohort to do the post-weaning test. Then we have to sacrifice the animals of the second cohort by 1 year old. And then we have to do the histological test and blood cytokine profiling to confirm our data of the behavioral test. But due to some limitations in our lab, we decided to start with the second cohort for the moment, and we are just now about finishing our post-weaning behavioral test, and we will sacrifice very soon our animals. So right now I will show you our behavioral test outcome from the second cohort. So as I mentioned, we did several reflex, we did development and neuromotor ability using test of writing reflex, negative geotaxis, and the cliff avoidance. So there was no significant effect with the writing reflex and negative geotaxis, but when it was the cliff avoidance, we showed that in females, the females in the treatment group achieved the criteria of this test later than the control, as you can see here. And then we started the post-weaning behavioral test. We started with a social interaction to see if there's an effect on the sociability. And here we found a sex effect. So when we analyzed the data from females, we have a significant effect on the sociability in the aluminum group, as you can see here in the aluminum group. So there is no significant difference between the time spent with the empty cage versus the stranger one. And here you can see that the vaccine, there's no significant effect when we use 3 times the dose with the vaccine. And when we analyzed the data of the males, we observed that the males were, the sociability in males were, or was more affected by the vaccine treatment, but not with the vaccine 3 and the aluminum 1. And when the criteria was the social novelty, so it's the preference to the familiar versus unfamiliar animal, we didn't have any sex-dependent effect, and we showed that the animal, or the social novelty of this animal was affected by the vaccine and the aluminum treatment, but not with the vaccine 3 treatment. And then we did the light dark box test to evaluate the anxiety-like behavior in mice. So we showed that we have an increase in the anxiety and decrease of the exploratory behaviors in all treated group. But we showed that the females were more affected by the vaccination as compared to males, and the same thing, we didn't have any significant effect between the vaccine 3 versus vaccine group. And then we did the novel object recognition test at the age of 6 to 7 months to evaluate the memory performance. And here, as you can see, we showed a memory performance alteration only in the aluminum group as compared to control and other treatment groups. And during our post-weaning test, we have this issue, the issue of barbering, which is the fur and whiskers eating. We had this issue between 4 to 10 months in all treatment group, and especially in females. As you can see here, in control, we didn't have any female barber as compared to vaccine and aluminum group. So to conclude all of that, our data showed that the animal undergo a late development of neonatal cliff avoidance reflex in females from all group versus control. And we can say that the vaccine 3 cannot be a proper positive control, which can be explained by the fact that we published when I was working at Professor Girardi's lab in Paris, that the non-dose dependent effect pattern of the particles of aluminum, and it will be the same thing. We can consider it in the same thing in vaccine particles as well here in our study. And we showed the increase of the anxiety and the decrease of the exploratory activities in all treatment group, which matches the previously published article by our team in Vancouver. And we showed the sociability deficit in aluminum and vaccine groups, but not in vaccine 3 group, which matches the very recently published article by our team in Canada, and the other article which very recently as well published by our friends from Spain. And we showed a sex effect here. So the sociability was affected in females by aluminum treatment, but in males the male were more affected by vaccination. And we showed the alteration of the recognition memory performance only in adjuvant group. So here our data are kind of shaping up to be 4x4 matrix, where the variables are age, sex, aluminum, and full vaccine injection. So leading to outcome vary between group and treatment. However, the behavioral data alone, especially from rodent, will not be enough to make inferences about human disorders. So we are waiting for the upcoming step about the blood profiling of cytokines and the histological data to just compare or confirm the data from the behavioral test and figure out what's going on. So just about somewhat about the barbering issue. To be honest, it was not one of our endpoints when we did the design of this study. It just showed up during our, when we were doing our post-weaning test. And in the literature, this barbering issue seen as a model of trichotillomania, which is seen in both impulse control and obsessive compulsive disorders. And this barbering was seen as well as disease of oxidative stress. And the Barber mice show a pattern of neuropsychological biomarkers that matches the TTM. In our study, the barbering was more important in females, especially from the vaccine and aluminum group as compared to control, which is in agreement somehow with the same study, the very recent study from our friend from Spain about sheep, the barbering in sheep as well. But the only difference, they observed this issue only in vaccine group, but not in the adjuvant one. Before I thank you for your kind attention, I have to thank my team in Vancouver, especially my boss, Professor Show, and I have to thank the CMSRI, especially Claire Dawson, for her kind support to our study. Thank you very much.
I can ask something because it's always intrigued me, and it relates to many other things that we've already talked about and will talk about, which is, do you have any feeling for why we are seeing such gender differences?
This is a big question, actually. I don't have any answer for this, for this question, because I think until now it's not just the gender as well, or the sex. So I think it's a very complex question, a very multi-factors question. So we cannot see this problem through the sex effect. So we have to take on consideration all of these factors, as I mentioned, the age, even though the strain. You remember very well, Roman, when we did the experiments with the aluminum adjuvant particles in CD1 versus black cis, there was a very huge difference between the translocation or the biodistribution of these particles in the butt of this strain. So we cannot take this sex or effect alone without the other factors. So, but again, for your question, I don't have any precise answer.
Thank you.
If you have a...
No?
I have a question. I did not check up the design of the study. You evaluated all the symptoms after the first, the second, the third vaccination, or just at the end?
No,
it's just a question because...
no, this window of age between 7 to 22 days, postnatal days, it corresponds to 0 to 18 months in children,
yeah.
More or less. And we did this vaccination according to the CDC schedule in the state. And then we started, in the same time we started the pre-weaning test, and then we continue with the post-weaning test.
And the vaccination was with all the 3 doses?
Sorry?
When you vaccinated?
Yeah.
With all 3 doses? I simplify the question.
Yeah.
In my practice, I see more events of the vaccine after the boosting, not the first. The first one goes well at 2 months. But at 4 months, when I have to repeat the vaccines, I observed much more events linked to the vaccine.
The same one?
The same vaccine.
Okay.
So
I don't have any answer. Maybe it's Yeah, I think there's an accumulative effect, but maybe it's the first one as well. You can see like later, not like right away.
Yeah.
But I don't know. Maybe Roman have an answer about this question.
Can I think the question was asking,
Yeah.
whether you administered the vaccine on, for example, 3 occasions to the mice,
Yeah.
or
this
whether
was-
you administered the full schedule in 1 dose.
Yeah.
And do you see any, and were there any indications that showed a difference between those methods?
Thank
So,
you.
is that right?
Yes.
Say it again, please. It's very complicated.
When you gave the mice
Yeah,
the vaccine times 3,
yeah.
whatever it was, the aluminium adjuvant on its own,
Yeah.
did you give it as a single dose, or did you give it as the schedule asks you to?
No, a single dose.
Okay, so
Okay. Thank you.
No, the vaccine 3. This is the idea of the use of this vaccine 3. We tried to have a positive control, but it was not a positive control actually due to the fact of the non-dose dependent effect. So in this case.
Yeah, and the last question about the behavioral, you said anxiety was increased and exploratory behavior decreased. Did you remark or observe some aggressivity?
Yeah, the barbering issue
Yeah.
can be seen like aggressivity as well somehow.
Can you tell me again what is barbering? issue?
Barbering, it's just eating the fur and the whiskers for other animals. You know, one of the cage mates can eat the fur and the whiskers for the other cage mate.
Oh,
See, so yeah.
so this makes me think that one child, which was vaccinated about 2 years old, and late, he had a chewing.
Uh-huh.
It was transitory, in a few weeks.
So
But
the-
could we have something?
the barbers, you know, the barber mice are seen like a model of the trichotillomania. So I mentioned it, but it's seen apart from the autism. It's not a model of autism or other neurodevelopmental diseases.
okay.
Thank you.
Thank you. We have time for one quick question.
Just one quick question. Is there any consideration to
Tell
move?
us who you are.
My name's Del Bigtree. I'm a nonprofit, the Informed Consent Action Network. I'm from the U.S. Is there any thought to move towards primates where there's more of a similarity to human beings now that we're seeing behavioral changes and things? Do you see any of this work going towards a primate study that would be similar? Or see an advantage to that?
No.
No. Okay. Thank you.
Okay, thank you.